Hello everyone, this is Saleem, your biology instructor, and welcome back to our biology class. Today we will learn about the very important discipline of science, biotechnology. Well, I have divided this discipline into two categories, this topic into two categories, the basic introduction and a very important part of biotechnology, genetic engineering. Well, moving on, what is biotechnology? Biotechnology is the use of living organisms in the process for the manufacture of useful products or for services. In simpler words, the use of one living thing for the benefit of another living thing is the major essence of biotechnology. Okay, biotechnology is not a new branch of science. It is a very old technique. Human began using microorganisms as early as 4000 BC for making wine, vinegar, cheese, yogurt, etc. Some of these processes have become a part of everyday life, that we even may hesitate to refer them as, biotechnology. But the fact and truth is, we have been making use of biotechnology since we discovered farming. And the basic techniques in biotechnology used by farmers is cross-pollination of plants and crossbreeding. They are very major techniques. These techniques were used to enhance product quality and to meet specific requirements. So we know that biotechnology is not a new discipline. The discipline is very old, and fermentation and other such processes which are based on natural capabilities of organism. These were referred to the old biotechnology. But genetic engineering, this is the new field of biotechnology. Moving on, biotechnology, as I have told and has stated earlier, using one living organism for the benefit of another organism is basically what biotech is used for, and this major thing is done using DNAs or RNAs, the genetic material. These things are done on genetic material. Moving on to the scope and importance of biotechnology. What is the effect of biotechnology? What are the applications of biotechnology? Number one, biotechnology in the field of medicine. In the field of medicine, biotechnologists synthesized insulin and interferon. If we consider the field of medicine, biotechnology has a vast range of applications. So, moving on, a large number of vaccines and antibodies, which include human growth hormone and other medicines, have also been produced. Various enzymes are being synthesized for medicinal as well as industrial use. Gene therapy, the basic therapy, this is basically the treatment using genes, has become important in recent years. Biotech has also proved much beneficial in forensic medicines and the study of DNA. Well, as we know, it helps in the identification of criminals, it helps in the identification of human bodies. Moving on, biotechnology in the field of food and agriculture, the second most important work of biotechnology. Well, fermented foods, the basic thing of food when we talk about food and agriculture. Fermented foods such as pickle, yogurt, malted foods. For example, powdered milk, a mixture of barley, wheat, flour, and whole milk. Various vitamins, dairy products. What are they? They are produced using microorganisms. So basically, they are all applications of biotech. And let's talk about foreign countries. wine, beer, they're all produced in beverage industry. How are they produced? They are produced by the use of biotechnology, by this discipline. This discipline helps them do this. The criteria of this discipline helps them do this. And in the field of food and agriculture, when we talk about this, we come across a word called transgenic. Transgenic organism. What are transgenic organisms? Organisms with modified genetic setup. They are transgenic organisms. They organisms that have been modified for our own use, so we may get the quality we need, the requirements we want. Those are transgenic. Transgenic may be plants or animals. Transgenic plants are being developed in which desirable characteristics are present, like more yield, resistance against diseases, insects, and herbicides. In fact, transgenic animals are also being produced. They give, well, we were talking about transgenic animals, so transgenic goats, chicken, cows, they give more fruit and more milk. Many animals like mice, goat, cows have been made transgenic to get medicines through their milk, blood, or in fact urine. They are very useful. But transgenic animals, transgenic plants, they are very useful. Okay, now moving on. Biotechnology and environment. What is biotechnology is doing for environment? Well, biotechnology is also being used for dealing with our environmental issues, like pollution control, development of renewable resources for energy restoration of degraded lands, and biodiversity conservation. So we know biotechnology is doing a lot of work in every field, whether medicine, food, agriculture, and for that purpose bacterial enzymes are used to purify water. Microbes are being developed to be used as biopesticides, biofertilizers, biosensors, etc. Such transgenic microorganisms, they are used for recovery of metals, cleaning of spilled oils, and for many other purposes. Biosensors are basically used for the recovery of metals. The microorganisms used for the recovery of, metals, etc. And as the advancements are taking place in this discipline, fears are also growing up that there might be, many advances in biotech in terms of release of harmful organisms which are, developed through recombinant DNA technology, which is also known as genetic engineering. The second very important discipline, the basic important category of this discipline is genetic engineering. Genetic engineering, often considered as the new form of biotech. Yeah, it's considered as new biotechnology. What is genetic engineering? Genetic engineering is artificial synthesis. It is modification, removal, addition, and repair of genetic material, DNA. This is the modern biotechnology. It is just done to, alter the characteristics of organisms. As you can see, this diagram, this shows that how genetic engineering is done, how DNA is cut, and how a plasmid is taken from an organism, the vector organism, and how it's transferred. This is how genetic engineering is done, which I'll explain further in this lecture. The work on genetic engineering, as I have told you the definition, the work on genetic engineering, it started in 1944 when it was proved that DNA carries the genetic information. Scientists isolated the enzymes of DNA synthesis and then prepared DNA outside cells, And in 1970s, they were able to cut and paste the DNA of organism. And in 1978, scientists prepared human insulin by inserting the insulin gene in bacteria. The process of production of human insulin will also be studied further in this lecture. Human growth hormone was also synthesized in bacteria. In 1990, a very important project known as Human Genome Project was launched to map all the genes in human cells, but it was completed in 2002. A complete map of human genome that was published in 2002. Genetic engineering is also termed as recombinant DNA technology, and we have studied the definition. Moving on and telling more about genetic engineering, which was developed in the mid 1970s. If moving on, how is it when genetic engineering was done and the cutting and pasting of DNA, this resulted that the characteristics of the host organism were changed. If host organism is a microorganism such as bacterium, the transferred DNA is multiplied many times as the microorganism multiplies. Consequently, it is possible to obtain millions of copies of a specific DNA inside a bacterial cell. What are the objectives of genetic engineering? There are many objectives of genetic engineering. Why is genetic engineering introduced? For the isolation of particular gene or a part of gene for various purposes such as gene therapy, a very important application of genetic engineering. For the production of particular RNA and protein molecules, improvement in the production of enzymes, drugs, and commercially important chemicals, production of varieties of plants or animals, transgenic organisms, and treatment of genetic defects in higher organisms. This is also a very, very important application or objective of genetic engineering. The treatment of genetic defects was achieved through this modern technique of biotech, the treatment of genetic defects, thalassemia, etc. We are talking about genetic engineering. Like, what are the steps involved in basic steps involved in genetic engineering? How is it basically done? Well, there are 4 to 5 major steps which are done in genetic engineering. The first step is the isolation of gene of interest. In the first step, the genetic engineer identifies the gene of interest in a donor organism. That what is required, what is the gene we are looking for. The special enzymes called restriction endonucleases are used to cut the identified gene from the total DNA of the donor organism. I'm repeating. The enzymes are used for the cutting of the identified gene, which are called restriction endonucleases. The second step involves insertion of gene into a vector where it will reproduce. A vector is selected for the transfer of the isolated gene of interest to the host cell. The vector may be a plasmid, the extra chromosomal DNA present in many bacteria. Plasmid is specifically for bacteria. Or a bacteriophage, the gene of interest is attached with the vector DNA by using endonucleases, breaking enzyme, and ligase, the joining enzyme. Well, the breakage is done by endonucleases, which was also discussed in step 1, and the joining enzymes are used, ligases, for joining the gene with the vector. The vector DNA and the dash gene of interest are collectively called recombinant DNA. What are recombinant DNA? The collectively the vector and the attached gene of interest are collectively called recombinant DNA. Thirdly, the transfer of recombinant DNA into host organism. The recombinant DNA is transferred to the target host. In this way, the host organism is transformed into a genetically modified organism. We would term it as GMO, the genetically modified organism. Fourth, growth of the GMO. The GMO are provided suitable culture medium for growth to give as much copies of the gene of interest as needed. Suitable environment is given for the GMOs, the genetically modified organisms that were made in the step 3, and thus they make copies, they reproduce, they grow, and the gene of interest is produced as much as needed. Fifth is the expression of the gene. The GMO contains the gene of interest and manufactures the desired product which is isolated from the culture medium. When we are studying genetic engineering, the basic example of genetic engineering includes human insulin production. Human insulin production. How is this done? The diagram shows how insulin is produced, how it is genetically engineered. This is engineered through a human cell. In a human cell, a piece of chromosome is taken, and with the help of endonucleases, the gene of insulin is isolated. On the other hand, in bacterium, the vector is taken that we would consider is plasmid. Plasmid is isolated, plasmid is cut, and now the gene of insulin isolated and the plasmid that is cut, they are both attached. The gene is attached with plasmid using ligase, and now the recombinant DNA is produced. The bacteria that takes up the recombinant DNA, and now it's called the genetically modified bacterium. Here, genetically modified bacterium will work as GMO, the genetically modified organism. A suitable culture medium is provided to this bacterium and it reproduces, and thus the production of insulin takes place, and the expression of gene, the GMO that contains the gene of interest, manufactures the desired product here, which is insulin. We will take it out. This insulin is basically taken out from the culture medium. Here the culture medium is the bacteria, so thus insulin is produced in a very simple manner. Moving on, now we will discuss about the achievements of genetic engineering, the very important part of genetic engineering. What have we achieved? What are the perks of genetic engineering? Well, there are various achievements of genetic engineering, which include the basic, which I have told, human insulin gene that is transferred to bacteria, and then insulin, the genetic modified bacterium became able to synthesize insulin. Diabetics are now receiving this insulin. The second is human growth hormone. In 1977, an E. coli bacterium was created. E. coli bacterium was created, and now this bacterium is capable of synthesizing this hormone. Third, thymosin. This hormone, thymosin, is basically an hormone which may prove effective against brain and lung cancers. This is also produced genetically by modified microorganisms. Beta endorphins. Endorphins, as we know, are happy chemicals, and beta endorphins are painkillers that are produced by the brain. They are also now manufactured and produced and synthesized using genetic engineering techniques. Fifth, genetic engineering produces a safe vaccine against the foot and mouth diseases. Similarly, many vaccines have been produced against human diseases such as hepatitis. So we know vaccines are now part of our daily lives, and they are basically produced using biotechnology's basic field, genetic engineering. Interferons are antiviral proteins that are produced by cells infected with viruses. In 1980, interferon was produced in the genetically modified microorganisms for the first time. Interferons, antiviral proteins, wow, they can also be achieved. They're also achieved by genetic engineering. Urokinase, the enzyme that dissolves blood clots, also the mercy of genetic engineering. So now there are just a few things of genetic engineering. There are many more, like hemophilia. We know that become possible to modify a gene in human egg cell. This can lead to the elimination of inherited disease like hemophilia. As I was talking about genetic therapy, hemophilia can be treated using gene therapy, the mercy of genetic engineering. Blood diseases, they can also be treated. Their thalassemia, sickle cell anemia, they result in single genes. They are the defects in single genes. Normal genes could be transferred into the bone marrow, and this could be cured. Genetic engineering have developed plants that can fix nitrogen directly from atmosphere. Such plants, they need less fertilizers. So we know that there are many mercies of genetic engineering. Genetic engineering has been playing a very important role in our life, and will always be doing so as the modern technology advances. Well, folks, this was all for today. I hope you enjoyed, because I did, and I hope you learned. See you all next time. Thank you so much, and don't forget to subscribe our YouTube channel Tutor5, and don't forget to watch our other lectures too. See you all around in the next lecture, in the next video. Take care, stay safe, stay home. Goodbye.